Good evening, students. Today, let us discuss a new chapter that is molecular basis of inheritance. The first topic is DNA, the genetic material. From 20th century or during the 20th century, the scientists started studying about the genetics. They did several experiments to understand what actually get transferred from the parents to their offspring. So they have similar characteristics of that of the parents. Several experiments were done, but the conclusive experiments which were widely accepted and which actually proved that DNA is the genetic material are done by Griffith, MacLeod, McCarthy, and the experiments given by Hershey and Chase. So, first of all, let us discuss the Griffith experiment or the transformation experiment. In the year 1928, Frederick Griffith performed a series of experiments on the bacteria Streptococcus pneumoniae. The Streptococcus pneumoniae bacteria are responsible for causing the disease pneumonia. For his experiments, he took two strains of Streptococcus pneumoniae, rough strain or the avirulent type, and the smooth strain or the virulent type. The smooth strain, they are actually surrounded by a capsule. This capsule is responsible for causing the disease pneumonia. So they are the virulent type, that is, they causes the disease. Whereas our strain, they do not possess the capsule. As a result, they cannot cause the disease. Hence, they are the avirulent type. So Griffith for his experiment took two strain, that is smooth strain or the virulent type, and R strain or the avirulent type. So in the course of his work, Griffith injected these strains into laboratory mice. When live S strain was injected into a live mouse, the mouse contracted pneumonia and as a result died of pneumonia. When live R strain or R virulent type was injected into the mouse survived. When heat killed S strain was injected into the mouse survived. The fourth mouse, when injected with a mixture of live R strain and heat killed S strain, found dead as a result of pneumonia. The fourth result was surprising to Griffith. When Griffith studied the fourth mouse, he found the presence of the R strain and S strain. Thus, he concluded that even after heating, certain substances in the S strain were not destroyed but transferred to the R type, enabling them to synthesize the capsule, making them virulent S type. This experiment is also known as Griffith effect, or most popularly known as bacterial transformation. Here, Griffith established that something was there that were transferred from the parents to the offspring, but what was it? He could not establish. His experiment was then again done by three scientists in the year 1933 to 44. Oswald Avery, Colin MacLeod, Maclean McCarthy. They actually proved that DNA is the genetic material. They proved that DNA from the hit-kill S cells had transformed the R strain of bacteria to S strain. In their experiment, they too took the two strains of Streptococcus pneumoniae, that is R strain, the avirulent type, and the S strain or the virulent type. In their experiment, they extracted all the organic substances from the hit-kill S strain, that is carbohydrate, proteins, lipids, DNA, etc. Then they introduced them in separate R strain culture plates. The R strain culture plate which received the DNA showed the transformation of R type to S type, whereas the rest they did not show any such transformation. Thus, they proved that DNA is the genetic material which was responsible for the transformation of R type to S type. The next experiment which proved that DNA is the genetic material was the blender experiment. This experiment was done by Alfred Hershey and Martha Chase in the year 1952. For their experiment, they took bacteriophage T2, a virus which infects Escherichia coli. Here we can see the diagram of a T2 phage. This is the DNA and this is the protein structure. Bacteriophage possesses DNA which alone possesses phosphorus, whereas the protein coat is made of amino acids methionine and cysteine which alone possess sulfur in their sulfur. In their experiment, they took bacteriophage bearing radioactive P32 DNA and bacteriophage bearing radioactive S35 protein coat. Then they allowed these bacteriophages to infect two separate Escherichia coli culture plates. After the infection, they are introduced into the kitchen blender, where the separation of the viral protein coat and the Escherichia coli took place. The bacteria are then introduced into the centrifuge, where the T2 phage and the infected E. coli are separated. On study, they found the presence of radioactive P32 DNA in the newly formed T2 phage. Thus, they proved that DNA is the genetic material. Thank you.